was very pleased by this invitation, I have to say, because to me there are really a lot of challenges, bioinformatics challenges in systematics today, and that's something that interests me a lot. As Tore said, I have some time, a part of my time I spend at Uppsala University still, till the end of this year or so. Some of the things I'm going to talk about, actually most of the things that I have been involved in that I'm going to talk about, is projects that are active in Uppsala at the moment, different students, other researchers. So, biology, we have terms for different things and we have definitions and they differ. And I don't know exactly how you define bioinformatics, but for me this is a convenient definition: the collection, organization and analysis of large amounts of biological data using networks of computers and databases. That covers what I think when I hear the word bioinformatics. And systematics you can define as the scientific study of the kinds and diversity of organisms and of any and all relationships. So that's quite a broad definition. But you can also say that there are some major components that make up systematics. And those are taxonomy, which is naming, describing and classifying organisms into a system of taxa. And one term that has been coined rather recently is biodiversity informatics, and I think that's the kind of informatics that should be applied to taxonomy. The other major part of modern taxonomy is phylogenetics, which is about the historical or evolutionary relationships among taxa, and there you could say as a subset of bioinformatics you could call that phyloinformatics. So, and I'm going to talk a bit about taxonomic data. In bioinformatics, I think most of you deal with large amounts of data and in the form of different kinds of molecular data, genomic data and stuff like that. And because we have got quite recently in biology tools to collect very large amounts of such data. But within the history of biology, we have other kinds of data that has been collected for a very long time. And taxonomy is one such discipline where such old data actually also often is very important today. In taxonomy, part of the word nomenclature, so the naming of taxa includes a lot of information. For example, Linnaeus, he named about 9,000 species in his time, now about 250 years ago. And today we have almost 2 million that has been described, actually even more if you count all synonyms. So that's a lot of information. And also those names are published in literature that is enormous and often quite difficult to access. The taxa has been classified in many different ways and continues to do so. Actually, you could say that every phylogenetic tree that is produced is a new classification also. And of course the taxonomist is dependent on study material, often in the form of museum specimens. Specimens that are deposited in natural history museums. Some of these specimens are more important than others, and those are called type specimens, and they are important because they ultimately define names, the names that we use. And there are other types of taxonomic information, you could say, like geographical distribution, morphological descriptions, and of course molecular and genomic data and other data that we need to summarize and present in some way. We also use taxonomy to identify organisms into taxa, and that can be in the form of interactive keys using, for example, morphological characteristics of the specimens, and recently also DNA sequencing, or perhaps not so recently, but the term DNA barcoding has been coined to signify the process of identifying taxa by DNA sequences. One difficulty if one wants to summarize or make a database model, for example, to store all this information, is the heart of taxonomy, the taxa themselves, because they are not concrete things. Oops. Because in phylogenetic systematics, taxa cannot be defined. They are hypotheses. So, and this means that there will never be a final taxonomy. We will have to live with conflicting taxon concepts, different classifications, regardless what people will tell you, that will be the case. If we don't reduce taxonomy or classification to what the word actually literally means, that is defining taxa based on some characters. Like Linnaeus, for example, in his famous sexual system, he classified flowering plants according to the number of sexual parts in the flower. And of course one can make such classification, but then we can also classify names or whatever. So if we want to view taxonomy with phylogenetic glasses, we have to live with this, and this is a make efficient databasing a bit difficult, which I will come back to. Okay, we have in my research group, I'm a plant systematist, and I'm interested in a particular group of flowering plants. And we have for many years now tried to store information during our work, and this has developed to a project where we like to use this database as a tool, as a workbench during work, but also something where we can publish the results. And as I said, taxa are the heart of taxonomy that are difficult to handle. But specimens are real. This is a real specimen that we can see, we can assign a number to it, and we can find it again in the shelf or whatever. And specimens are, of course, important when making hypotheses about taxa. We study a lot of specimens to do that. But we also have the nomenclature to name the taxa. And therefore we also have this link between specimens and nomenclature, because some of specimens are types linked to names, and then we use those names and assign them to taxa. As a molecular systematist, which is something almost all molecular taxonomic labs do, more or less now, is to use DNA sequence data. So we have lab protocols, and we get some DNA sequences from which we can derive gene phylogenies, and from those we derive hypotheses about taxa. And an important part of taxonomy is purely descriptive, and I will give you two examples of applications of how descriptive data can be presented from this tool. Yeah, so this is just an image from the database where you can browse through different buttons and get different kinds of information. X303 is an incarnation of Prometheus, which has been developed mainly by Mikael Tollesson at Uppsala University. Some of you might know. He also made his PhD here almost 10 years ago in zoology. And that's a database implementation. Prometheus is a descriptive model from the Napier University in Edinburgh, and it's intended to be used for making formal descriptions of taxa, morphological descriptions, that is. And I think this is an important enterprise, because such descriptive data, we have a lot of such, very much such descriptive data in taxonomy, but not in a very formalized way. And the Prometheus model is quite nice in the way that it formalizes this in a way that each description element consists of 3 parts. That is a structure, the property, and a value. So, for example, in a flower, a petal can have a property that is a color, and that color could be, for example, yellow. And in order to accomplish this, one needs to define a vocabulary including those structures, properties, and allow values. And this has been done for angiosperms, for example. And from an ontology, one can derive a template. Because if I want to describe roses, for example, I don't need all the vocabulary needed for all flowering plants. And of course the descriptions themselves that has explicit values for taxa or specimens. So in a flowchart view, this looks something like this, that we have the ontology from which we derive templates, where we make the description, and there from the descriptions we can also derive natural language descriptions, which may be nice. We can also derive determination forms from the templates, which we can use for identification. So this is an example showing that the structures often occur hierarchical, which is nice. So an entire plant, for example, can have the property lifespan that can be different kinds. But the entire plant also consists of stems and leaves, for example. And the leaf consists of petioles, blade, nucleus, and so on. But some elements doesn't fit into this hierarchy, and that causes problems. But this has been solved by this model by introducing general structures and regions. So, for example, a stem may have different regions, a basal region, for example. And the basal region might be hairy, but hairs may occur anywhere on the plant. So, when the information, the description are made, the values are entered, we get description like this that can be transformed into natural language description like this. And in this way we can present descriptions one by one instead of waiting for example, if taxonomic revision may include hundreds of taxa, and instead of waiting for all of them to be completed, we can publish them one by one, which I think is a great advantage. Another application that is linked to our database now is something called taxamapper, which is intended to display geographical distribution of taxa based on phylogenetic trees. But it can also use online data that are now available in different databases and can be used then for visual inspection of geographical distribution. So in this case we have a link to our research project database, but we also can use global occurrence databases like the GBIF, which stands for Global Biodiversity Information Facility, which was launched something like 5 years ago, and it's a very large international initiative that strives to database all specimens in natural history collections. Quite an ambitious project. So. But you could say it's yeah, okay. And we also have a global taxonomic database like the GPC, the Global Plant Checklist, which is part of the new species Plantarum project. The neo species Plantarum was the starting point for today's nomenclature. And this is an ambitious project of making an update of a global, not only a checklist, but also a descriptions of all plant taxa on Earth. And this distribution then can be displayed as specimen data, where each dot represents specimen, or as taxon distribution, where the Earth is divided into regions, and a region which is shaded here indicates occurrence of this taxon. Again, note the difference here between specimen and taxon, where specimens pertain to physical things, physical collections, whereas taxon data does not, in a direct way. So this is from a taxonomic revision that a PhD student did recently, Frida Egens, where this can be used to display the geographical distribution of the specimens that were used for deriving a gene phylogeny here from this particular group. And just to make a comparison, when we look for the names that we here used our local database, which we use for this taxonomic research, to what we find in when we look for the same names in GBIF, we get this obviously erroneous single specimen. So there are several large initiatives to summarize this. Species Plantarum was one, and there are even more ambitious projects which are intended to cover all life. Something called Species 2000, or the Catalogue of Life. And just a few days ago, a new project which is very heavily funded apparently, was announced, the Encyclopedia of Life. And in principle you could say they aim at summarizing all this taxonomic information that I listed previously on in a web interface. Unfortunately, and which has been criticized a lot from the taxonomic communities, that they are almost devoid of content so far. And I think I mean, they are doing certainly very nice things, and there are developments going on there, I'm sure about that. But I think the problem is to make people publish the data. How I mean, you need something that looks good in your CV, and I don't know what this is. Yeah. So we need to look a lot more at how to efficiently publish such data and how to get credit for it. So, next thing I want to talk about here is DNA barcoding, which you can define as taxonomic identification by use of DNA sequences. And this is obviously something that requires reliable taxonomies and well annotated sequences. You need a database of sequences from a particular region that can be obtained from the organism that you want to identify. And here, which is quite nice, I think, is that you can, using a piece of sequence, you can put that sequence into a phylogeny. So you can make phylogenetic rather than species identifications. And now I will present some work of Henrik Nilsson, who's sitting here in this room, because I think this is some very nice work which has been performed by the mycological group here in Göteborg, which I'm not part of any of this work, but I hope to be able to present it in a fair way. So, ectomycorrhiza are fungi that covers roots of many of our plant species. And they are very difficult to identify morphologically. So, given that we have sequences taken from fruiting buddings from those fungi, we may obtain a phylogenetic tree like this. And in the fungal community, the region of choice is the ribosomal DNA internal transcribed spacer regions, which is nice because they are very easily obtained also from herbarium material. Because the ribosomal DNA occurs in large amounts in the nucleus. There's a lot of variation within those sequences, also within species there's a lot of variation, so they are informative. And there are quite many such sequences already available. So, yeah, you could sequence those ectomycorrhiza fungi and make a blast search and find something that fits very nicely. But that will not always be the case. Sometimes your match will be annotated like fungus, or uncultured ectomycorrhizal fungus, maybe a bit better, and so on. Or even worse, you may get perfect fit to several different, obviously very unrelated taxa. So here something must be wrong. So for fungi, there are ITS sequences available for less than 1% of all known fungal species. And more of 25% of those are unidentified to species level. And in addition, up to 20% of those annotated with a species name are misidentified. So the mycologists here in Göteborg and some other have coined a new database called UNITE, which doesn't contain by far as many sequences as GenBank does, but they are of very high quality. The determinations are made by taxonomic experts, and all sequences are linked to voucher specimens, and often also illustrations, descriptions, and ecological information. The database is available openly, but the submission of data is restricted to experts. And there are some novel improved tools in addition to BLAST. So this is some picture from the database. And here you can also put your submit the sequence and get a phylogenetic tree back, like this. But sometimes you may get your sequence might not give a good match at all. So what will you do? Sit and wait for it to appear, or you can use another software developed by Henrik here, Emerencia, which watches all unidentified fungal ITS sequences weekly and looks for new matches. Another very nice tool here is MORE, which is you can say an updated approximate phylogenetic reference for Basidiomycete. So in MORE, the sequences not only are BLAST and not only search for matches, but also aligned and analyzed phylogenetically, so that you will get an updated tree also with the support values. I think these also are jackknife percentages. All right. So different clades are defined, so you can also directly assign where the particular novel sequence, for example, will appear, to which higher taxonomic group it belongs. Okay, finally, I'm just going to say a few words about bioinformatics also, which is, you could say, phylogenetic analysis of large amounts of DNA sequences and other genomic data, and that was just what I showed in the previous slide. So these taxonomy and phylogenetics are obviously tightly linked. And in phylogenetics, there is a large number of methods available now, and I'm not going to dwell on that at all. But I just want to emphasize one thing which has interested me in the past or recently, and that is that phylogenetic relationships, they can be displayed either by a tree or by a network. And a tree is mathematically something that doesn't make cycles, that makes new daughter branches that never go together again. And that's how we usually look on phylogenetic relationships. But phylogenetic relationships can also be reticulate. That is, we might have a hybridization, for example. And then a network is the conceptual model. But at some level, it is convenient to use a tree model, ultimately at the single nucleotide level. There must be a tree. And such trees can be used to deduce trees or relationships at a higher level. And one problem here is, again, the difference here between taxa and specimens. Because species trees are quite simple in the sense that they are labeled by taxonames, and they occur only once. That is, if we make a phylogenetic tree from the properties of FATSA, this is how in the early days of phylogenetics, how phylogenetic trees were built usually. Whereas in molecular systematics or molecular phylogenetics, we almost always use gene trees, and often genes may occur in gene families that are not possible. So it's not possible to take apart the different members of the gene family a priori. So in a gene tree, the names may occur more than once. Okay. I think I have to finish it. But there are a number of processes that make gene trees to differ from each other, listed here. And so obviously we need some ways to identify those processes from each other. And often such processes are appropriately modeled by networks. And there are different approaches to this. There are distance-based approaches, and there are character-based methods, and there are tree-based methods. That is what I just was talking about, using several trees and combining them into a network that explains relationships at a higher organizational level. And this is an example from plants. Plants are allopolyploidy is an important process in plants. Allopolyploidy is a process where the gene of two parents are effectively added to each other to a hybrid that may make a new lineage. So, for example, here this is a diploid. 2x signifies a diploid, whereas 4x signifies a tetraploid, something that has four plant that has four instances of each chromosome. And as you can see here, this is a multi-tree where some of the names occur more than once. So, but this tree can be using an algorithm developed by Katarina Huber and Vincent Moulton, be converted to a network like this, which also concurs with chromosome, the allopolyploidization events in this group. So this tree that you saw in the previous slide is actually, you can unfold it from this network and you get exactly that. Okay, so finally, I think the challenges for systematics is very much collection and synthesis of data, and a lot of to ensure the quality of the basic data. So basic taxonomic research has to be funded, but taxonomists often complain about it. Oh, you just give money to molecular systematics and to bioinformatics and so on, but there's no content. And partly they're right, but the taxonomists must also organize the taxonomic data in efficient and available databases, available both for publication and data retrieval. And of course, phylogenetic analysis in the form of trees and networks, there's a lot to do given the really vast amount of information that we are facing. So finally, I just want to acknowledge the people that have been involved in producing most of the things that I've been showing here. Anja Rautenberg, PhD student in Uppsala, who has been working with the box database, Michael Tollesson, X303, Anders Larsson, Taxamaster, and of course Henrik Nilsson and his colleagues here in Göteborg called Unite Emergency and Board. And also Katarina Huvud, Vincent Moulton, and Martin Lott, the University of East Anglia, that have been developing methods for making networks. Okay, thank you.